Hello everyone, this is a study of Section 2, Regulation of Cell Cycle. A successful cell division cycle requires the orderly and unidirectional transition from one cell cycle phase to the next. For intrinsic regulator pathways, they are responsible for the precise ordering of cell cycle events. For example, entry into mitosis must always come after DNA replication. They also act as a clock or timer that turns on each event at a specific time, which provides a fixed amount of time for the completion of each event. It is a mechanism to ensure that each event is triggered only once per cycle in a complete irreversible fashion. Adaptability is necessary so that the system's behavior can be modified to suit specific cell types or environmental conditions. Backup mechanism is needed to ensure that the cell cycle can work properly even when parts of the system malfunction. For example, errors in the spindle assembly checkpoint can lead to chromosomal imbalance and aneuploidy, a feature characteristic of virtually all cancers. So, misregulation of proteins that control this checkpoint has been detected in human cancers. The cell cycle control system is based on two families of proteins, the specialized activating proteins cyclins and the cyclin-dependent protein kinase CDK. CDK can selectively phosphorylate downstream proteins on serine and serine residues. Cyclins bind to CDK molecules and control their ability to phosphorylate target proteins. They can undergo a cycle of synthesis and degradation in each division cycle of the cell. Cyclin-dependent kinase inhibitors CDKs can selectively inhibit the kinase activity of CDKs by binding to complexes of CDKs and cyclins such as protein 21, protein 27, and protein 16. Cell cycle progression is governed primarily by inhibitory regulation. Anaphase promoting complex APC is a complex of several proteins which is activated during mitosis to initiate anaphase. Cyclins are among the most important core cell cycle regulators. Cyclins are a group of related proteins, and there are four basic types found in humans: G1 cyclins, G1S cyclins, S cyclins, and M cyclins. For example, cyclin D as a G1 cyclin, cyclin E as a S cyclin, cyclin A as a S and mitotic cyclin, cyclin B as a mitotic cyclin. As the names suggest, each cyclin is associated with a particular phase, transition, or set of phases in the cell cycle and helps drive the events of that phase or period. For instance, M cyclin promotes the events of M phase, such as nuclear envelope breakdown and chromosome condensation. The levels of the different cyclins vary considerably across the cell cycle. A typical cyclin is present at low levels for most of the cycle, but increases strongly at the stage where it's needed. M cyclin, for example, peaks dramatically at the transition from G2 to M phase. Cyclins drive the events of the cell cycle by partnering with CDKs. A lone CDK is inactive, but the binding of a cyclin activates it, making it a functional enzyme and allowing it to modify target proteins. There are three main CDKs: CDK4 for G1 phase, CDK2 for S phase, CDK1 for mitotic stage. In them, the complex of CDK1 and cyclin B is also called mitosis promoting factor MPF, which is a maturation promoting factor. How is cyclin-CDK complex regulated in cell cycle? CDKs are kinases, enzymes that phosphorylate specific target proteins. The attached phosphate group acts like a switch, making the target protein more or less active. When a cyclin attaches to a CDK, it has two important effects. It activates the CTK as a kinase, but it also directs the CTK to a specific set of target proteins, once appropriate to the cell cycle period controlled by the suckling. For instance, GYS sucklings send CTKs to S-phase targets for promoting DNA replication, while M sucklings send CTKs to M-phase targets for making the nuclear member break down. Sucklings and CTKs are very evolutionarily conserved. In general, CTK levels remain relatively constant across the cell cycle, but CTK activity and target proteins change as levels of the various sucklings rise and fall. In addition to needing a suckling partner, CTKs must also be phosphorylated on a particular site in order to be active, such as serine 161 in CTK1, and may also be negatively regulated by phosphorylation of other sites such as serine 14 and tyrosine 15 in CTK1. Phosphorylation around T160 by CTK activating kinases CAKS improves substrate binding and complex stability to enable full CTK activation, whereas phosphorylation at T14 and Y15 within the ATP binding site by inhibitory kinases V1 and MIT1 interferes with proper ATP alignment. Whereas most sucklings promote CTK activity, CKIs retain CTK activity. CKIs are subdivided into two classes based on their structure and CTK specificity. The INK4 family members, including P16INK4A, CDKN2A, P15INK4B, CDKN2B, P18INK4C, CDKN2C, and P19INK4D, CDKN2D, primarily target CDK4 and CDK6. Conversely, the CYP-KIP family members, including P21CYP1, CDKN1A, P27KIP1, CDKN1B, and P157KIP2, CDKN1C, are more promiscuous and broadly interfere with the activities of suckling D, E, A, and B dependent kinase complexes. Cell cycle control system depends on cyclical proteolysis, SCF enzyme complex responsible for the ubiquitination and destruction of GYS sucklings and certain CKIs and control S-phase initiation. CKI protein can be phosphorylated for the binding and multi-ubiquitination of active SCF for proteasome degradation. APC in M-phase is responsible for the ubiquitination and proteolysis of M sucklings and other regulators of mitosis, which is triggered by MPF. In MPF, M suckling stays at low levels for much of the cell cycle, but builds up as the cell approaches the G2M transition. As M suckling accumulates, it binds to CTKs already present in a cell, forming complexes that are poised to trigger M-phase. Once these complexes receive an additional signal, essentially an all-clear confirming that the cell's DNA is intact, they become active and set the events of M-phase in motion by phosphorylating various proteins, which peaks during metaphase. Then, MPF triggers its own destruction by activating the APC, a protein complex that causes M sucklings to be destroyed, starting in anaphase by ubiquitination and proteasome degradation. The inactive APC is activated by the binding of activating subunit CDC20. The destruction of M sucklings pushes the cell out of mitosis, allowing the new daughter cells to enter G1. APC also causes destruction of the proteins that hold the sister chromatids together, allowing them to separate in anaphase and move to opposite poles of the cell. The Nobel Prize in Physiology or Medicine 2001 was awarded jointly to Leland Hartwell, Tim Hunt, and partners for their discoveries of key regulators of the cell cycle. They identified the molecules CDK and sucralin that control the cell cycle. This is the end of the section. Thank you very much.